everyone. Today we will discuss about development of nervous system. The objectives are to learn about the formation of the neural tube, the neural crest cells, development of spinal cord, development of brain, the flexures of the brain and the clinical aspect. The whole of the nervous system is derived from the ectoderm except its blood vessels and some neuroglial elements. So, the specific cell population of early ectoderm which gives rise to the entire nervous system and special sense organs is termed as neural ectoderm. The neural ectoderm later differentiates into three structures, that is neural tube, neural crest cells and ectodermal placodes. The neural tube gives rise to the entire central nervous system. The neural crest cells form the entire peripheral nervous system and the ectodermal placodes contribute to the cranial sensory ganglion, hypophysis and the inner ear. So, we have seen the central nervous system is developed from the walls of the neural tube. So, here you can observe that circular structure which is blue in color. So, that is the neural tube. So, that is giving rise to the entire central nervous system, and the neural crest cells here it is represented by green color. So, they gives rise to the peripheral nervous system. Now, we will see the neural tube formation. So, in the early embryonic disc, that is at about 16th day of embryonic life, the ectoderm overlying the newly formed notochord thickens in the midline, forming the neural plate. So, initially the neural plate is straight or flat and peripherally it is narrow. The somatic mesoderm develops on either side of the notochord. So, the margins of the neural plate are elevated as the neural folds. So, here we can observe the purple color thing which is above the neural groove, that is called the neural folds. As a result, the center of the plate sinks, creating the neural groove. So, the neural folds gradually move towards the midline and finally fuses to form the cylindrical structure that is called the neural tube. So, here we can observe the cylindrical structure which represents the neural tube. So, this neural tube loses its connection with the surface ectoderm. So, the process of this formation of the neural tube is termed as neurulation. So, this fusion of the neural folds begins in the middle, that is the region of 4th somite on the 20th day of embryonic development, and it proceeds simultaneously towards the cephalic and caudal directions. So, it occurs at about 22nd day after conception. So, the fusion of the cranial and the caudal ends of the neural tube is delayed, forming a small openings called the anterior neuropores. So, here you can observe, and the posterior neuropores. So, the neural tube and overlying amniotic cavity remain temporarily in open communication with each other through these ports. So, the anterior neuropore closes earlier in the middle of 4th week at about 18 to 20 somite stage, that is about on the 25th day, and the posterior neuropore closes later at the end of 4th week at about 25 somite stage, that is on 27th day. So, by the time neural tube is completely closed, it is divided into an enlarged cranial part and the elongated caudal part. So, that will be developing in future as a brain and the spinal cord respectively. So, if there is a failure of anterior neural tube closure, then it leads to the anencephaly. There will be a degeneration of the forebrain and the skull. If there is a failure of the posterior neural tube closure, then it will lead to the spina bifida. So, here you can observe in this diagram, that is normal and the anencephaly and the spina bifida. If we supplement the folic acid during pregnancy, these complications can be avoided. So, the folic acid, it plays an important role. So, the anencephaly means the failure of the cephalic part of the neural tube to close, and it is associated with the defective vault of the skull. The features includes the vault of the skull is absent, the brain is represented by a mass of degenerated tissue exposed to the surface, the cord is open in the cervical region, then the prominent eyes bulging forward, and the chin is continuous with the chest due to absence of neck. The spina bifida. So, we have seen an incomplete closure of the caudal neuropore. There will be a defective development of associated vertebral arches. The features of the spina bifida includes the failure of dorsal portions of vertebral arches to fuse each other, usually localized in the lumbosacral region, and the neural tissue is widely exposed to the surface. The neural tissue shows considerable overgrowth. So, here in this picture you can see the spina bifida, which is open defect, and you can observe the dura mater and the spinal cord, it is propping out. Now, the formation of neural crest cells. So, as the neural folds comes together and fuse, cells at the tips of neural folds break away from the neuroectoderm to form the neural crest cells. So, the surface ectoderm of one side becomes continuous with the surface ectoderm of opposite side over the neural tube. So, thus cells at the tips of the neural folds, so that forms the neural crest cells. They do not participate in the neural tube formation. The neural crest cells at first remain in the midline between the dorsal surface of the neural tube and the surface ectoderm, and then forms the two cell clusters dorsolaterally, one on either side of the neural tube. So, here you can observe the centrally the neural tube is there, on either side the neural crest cells are distributed. So, these neural crest cells differentiate to form the different structures that includes the melanocytes, the cells of posterior root ganglia and the ganglia of cranial nerves, the cells of autonomic ganglia, cells of adrenal medulla and the Schwann cells. So, these are the derivatives of the neural crest cells. Now, the formation of ectodermal placodes. So, prior to the neural tube closure, the neural fold contains two types of cell populations, that is neural crest cells and neuroepithelial cells. So, during neurulation, the neural crest cells are detached and neuroepithelial cells gets incorporated into the surface ectoderm. These areas of neuroepithelium within the surface ectoderm are termed as ectodermal placodes. Now, the development of the spinal cord. So, the spinal cord develops from the caudal elongated part of the neural tube. So, we have seen the cranially enlarged part is there, that is giving rise to the development of the brain, whereas the caudal elongated part gives rise to the spinal cord. So, the cavity of the tube constricts to form the spinal canal. The grey matter of the spinal cord as butterfly shape. Upper part is give rise to the dorsal horn. The lower part the ventral horn, and in between these two, that is the intermediate zone is present between the dorsal and the ventral horn. So, the cells of this region, that is when you take a cross section, the cavity of the neural tube appears like the vertical slit. So, that will be observed as a central canal. Due to the formation of the vertical slit, the dorsal and the ventral walls of the neural tube remain thin and are called roof and floor plates. The lateral walls of the neural tube gets thickened. On each side, the lateral wall of the neural tube is demarcated into dorsal and ventral regions by the inner longitudinal sulcus called the sulcus limitans. The cells of the dorsal region, that is called the alar lamina, are functionally afferent or sensory, whereas those of the basal lamina leave the cord as the efferent or the motor. So, the axons of the cells of basal lamina leave the cord as ventral roots and join with the peripheral process of dorsal root ganglia to form the spinal nerves. So, the cells of the alar and the basal laminae are arranged into longitudinal columns. Each lamina reveals two columns. That is, the two afferent columns of alar lamina receive the axons from dorsal root ganglia. That includes the general somatic afferent column. It extends throughout the spinal cord and receives the impulses from the superficial, that is the cutaneous, and the deep, that is proprioceptive receptors. The general visceral afferent column, it is confined to thoracolumbar and the sacral regions only and receives the impulses from viscera and the blood vessels. The two efferent columns of the basal lamina give rise to motor fibers. Those are the general visceral efferent column. It is confined only to the thoracolumbar and sacral regions and provides preganglionic fibers to viscera, glands and blood vessels. Then the general somatic efferent column, it extends throughout the spinal cord and provides fibers that innervate the skeletal muscles. So, the white matter consists of the columns of axons, that is dorsal column, the lateral columns and the ventral columns. Now, the positional changes of the spinal cord. At week 8, the lengths of the spinal cord and the vertebral column are equal. The spinal cord extends along the entire length of vertebral canal and the spinal nerves exit the intervertebral foramina at the level of their origin. Due to the differential growth of the cord and vertebral column, the intervertebral foramina do not remain at the level of spinal nerves. So, in order to exit from the corresponding intervertebral foramina due to the recession of the spinal cord, the spinal nerves are forced to go down in the oblique direction. At week 24, the lower end of the spinal cord ends at the S1 vertebrae. At birth, the lower end of the spinal cord ends at the level of L3 vertebra, and in adults, due to the further recession of the cord, the lower end of the spinal cord ends at the lower border of the L1 vertebra. The nerve roots, that is lumbar, sacral and coccygeal nerve roots that descend below the lower end of the spinal cord, that is conus medullaris, surround the thin thread like prolongation of the pia mater, forms the tip of the conus medullaris, that is the phylum terminale, and form the cauda equina. Now, the development of brain. The brain develops from enlarged cranial part of the neural tube. At about 4th week, the enlarged cephalic part of the neural tube shows 3 distinct dilatation called the primary brain vesicles. These are the prosencephalon, then the mesencephalon and the rhombencephalons. Their cavities form the ventricular system of the adult brain. So, during 5th week, both prosencephalon and rhombencephalon subdivide into 2 vesicles and forms the secondary brain vesicles. So, now the flexures of the brain. So, the primitive brain presents 3 flexures, that is the pontine flexure at the middle of the rhombencephalon, the cervical flexure at the junction of rhombencephalon and the spinal cord. So, this flexure is convex dorsally and appears at the junction of the hindbrain and the spinal cord, making a right angled bend between them. Then the cephalic or the mesencephalic flexure in the region of the midbrain. It is convex dorsally and appears at the midbrain level. Now, the development of the ventricular system. The cavities of the brain vesicles form the ventricular system of the adult brain. The hindbrain cavity becomes the 4th ventricle. The diencephalic cavity becomes the 3rd ventricle. So, the narrowed mesencephalic cavity becomes the cerebral aqueduct between the 3rd ventricle and the 4th ventricle. So, the cavities in the lateral, that is cavities in the cerebral hemisphere becomes the lateral ventricle. So, thus the brain consists of 4 ventricles, that is 2 lateral ventricles, a 3rd ventricle and the 4th ventricle. The 2 lateral ventricles communicate with the 3rd ventricle via interventricular foramen. The 3rd ventricle communicates with the 4th ventricle through the cerebral aqueduct, and the 4th ventricle is continuous below with the central canal of the spinal cord that presents a small dilatation at the inferior end called the terminal ventricle. So, if there is a obstruction in the flow of CSF or increased production of CSF, that leads to the hydrocephalus. So, it is the hydrocephalus is the condition characterized by the dilatation of the ventricles. The dilatations may be due to excess accumulation of CSF or due to overproduction of the cerebrospinal fluid or obstruction to its circulation. So, here we can observe the normal ventricles and the enlarged ventricles in the hydrocephalus. The features includes disproportionately large size head, the setting sun appearance of the eyes. Dandy-Walker's syndrome. It occurs due to atresia and blockage of apertures in the roof of the 4th ventricle, and it is characterized by dilatation of the 4th ventricle, agenesis of cerebellar vermis, occipital meningocele, and the agenesis of splenium of corpus callosum. So, now we will learn about the prosencephalon or the forebrain. So, the diencephalon develops from the median portion of the prosencephalon, and the upper enlarged part, that is the telencephalon, gives rise to telencephalic vesicles that will give rise to the cerebrum. Then the diencephalon also gives rise to the optic vesicles. So, here we can observe the optic cup, then the optic stalk. Later it develops into the development of the eye and the optic nerve. So, here we can observe the telencephalon giving rise to the 2 cerebral hemispheres. So, the developing cerebral hemispheres enlarges forward, upward and backward in that order. As the vesicle grows backwards, it overlaps successively the diencephalon, mesencephalon and cerebellar rudiments. So, it lie over and lateral to the diencephalon. Then the lowest parts of the medial walls of hemispheres in the region where they are attached to the roof of the diencephal remain very thin due to disproportion growth of the various parts of the hemispheres. So, at the inferior aspect of the frontal region of the cerebral cortex, we can observe the olfactory bulbs and related structures that participate in the sense of smell. Then the cells of the walls of the telencephalon divide and differentiate into various structures. So, that is white matter systems develop carrying axons to and from the neurons of the telencephalon. The fluid filled spaces within the cerebral hemispheres becomes the lateral ventricles, and the space at the center of the diencephalon becomes the third ventricle. The walls of the telencephalic vesicles appear swollen due to the proliferation of neurons. So, in the upper part it gives rise to the cerebral cortex, and in the lower part it gives the basal telencephalon. So, in the basal telencephalon there will be a presence of sulcus, that is a hypothalamic sulcus, which will divide and gives rise to the thalamus and the hypothalamus. The neurons of the developing forebrain extend the axons to communicate with the other parts of the nervous system. These axons bundle together to form three major white matter systems. The cortical white matter contains all the axons that run to and from the neurons in the cerebral cortex. The corpus callosum forms an axonal bridge that links the cortical neurons of two cerebral hemispheres. So, here you can observe the corpus callosum bridging the two cerebral hemispheres. Then the dorsal surface of the mesencephalic vesicle becomes the structure called the tectum. The floor of the midbrain forms the tegmentum, and the cerebrospinal fluid filled space in between the constricts into a narrow channel called the cerebral aqueduct. So, the aqueduct connects rostrally with the third ventricle of the diencephalon. So, the hindbrain or the rhombencephalon differentiates into cerebellum, pons, medulla, the CSF filled tube that gives rise to the fourth ventricle, and the continues with the cerebral aqueduct of the midbrain. So, the tissue along the dorsolateral wall of the tube, that is the rhombic ellipse, grows dorsally and medially until it meets the opposite side and thus forms the cerebellum. The ventral wall of the tube forms the pons. The ventral and caudal half of the hindbrain swell and covered only with the thin layer of non-neuronal ependymal cells. Along the ventral side of the medulla, these runs forms the medullary pyramids. So, the caudal part of the myelencephalon has a central canal and forms the closed part of medulla oblongata. Rostrally, the central canal expands as a cavity of the fourth ventricle, and thus the rostral part of myelencephalon forms the open part of medulla oblongata. So, the floor of the fourth ventricle is derived from the myelencephalon and metencephalon. So, that will differentiate to medulla and the pons. On either side of the midline, the floor consists of basal and alar lamina that separates from each other by longitudinal sulcus, that is sulcus limitans, as seen in the spinal cord. So, here also the nuclei are arranged afferent and efferent, just like in the spinal cord. In the spinal cord, each lamina consists two columns, somatic and visceral. But in the brainstem, to supply the derivatives of bronchial arches that develop around this region, the special bronchial column appears between somatic and visceral columns of each lamina. Thus, in brainstem, the basal lamina contains 3 columns and alar lamina contains the 4 columns. So, that includes the functional column with the basal lamina of the brainstem, the somatic efferent, the special visceral efferent, the general visceral efferent. The functional column in the alar lamina of the brainstem, general visceral afferent, the special visceral afferent, the general somatic afferent and the special somatic afferent. So, all the motor nuclei of the brainstem are derived from functional columns of its basal plate and all the sensory nuclei from the functional columns of the alar plate. So, the stretched roof plate of rhombencephalic vesicle forms the roof of the fourth ventricle. So, the roof consists of a single layer of ependymal cells covered by a vascular mesenchyme, the pia mater. The pia mater along with the covering layer of ependymal cells forms the tela choroidea. So, due to active proliferation of vascular mesenchyme, the tuft of capillaries of blood vessels invaginates into the ventricular cavity. So, this consists of tela choroidea and tuft of capillaries that forms the choroid plexus. So, we will just revise the development of the brain. It is having three primary vesicles, that is prosencephalon that give rise to the forebrain, mesencephalon midbrain and the rhombencephalon hindbrain. The prosencephalon again gives rise to telencephalon and diencephalon. The mesencephalon gives rise to the mesencephalon. The rhombencephalon gives rise to metencephalon and myelencephalon. The telencephalon gives rise to the cerebrum. The diencephalon gives rise to the optic cup, the thalamus, hypothalamus and epithalamus. The mesencephalon gives rise to midbrain. The metencephalon of rhombencephalon gives rise to pons and cerebellum. The myelencephalon part of the rhombencephalon gives rise to medulla oblongata. So, now the questions for assignment. Describe the neural tube formation and associated anomalies of the neural tube. Mention the derivatives of the neural crest cells. Thank you.